Everyone, so this week, week 8, we are going over the lymphatic and immune systems, and then we will do roughly half of the lecture on the heart, and then we'll continue the heart next week. So keep in mind too that many of you, maybe not all, but many of you will end up going on to take microbiology. So just know that you're getting sort of a cursory introduction to both the immune and lymphatic systems in A&P2, and then you'll go on to dive much deeper into the immune system in microbiology. So our immune system is the complex collection of cells and organs that works to both destroy and in some cases neutralize pathogens, right, microorganisms that can cause us disease or death. The lymphatic system is a system of vessels as well as certain types of cells and specific organs that carries excess fluids to the bloodstream and then filters pathogens from the blood. The swelling of our lymph nodes during an infection and the transport of lymphocytes, white blood cells, as we'll learn, via these lymphatic vessels are just two examples of the many connections between the immune and the lymphatic systems. Now, if we look at functions of the lymphatic system to start, blood pressure causes leakage of fluid from the capillaries, right? The smallest of the blood vessels, they're only a single cell layer thick, and it's simple squamous epithelium. So just with a little bit of increase in blood pressure, just normal fluctuations, fluid will leak from the capillaries, resulting in the accumulation of fluid in the interstitial space, which the interstitial spaces are the spaces between the individual cells in the tissues. In humans, roughly 20 liters of plasma, so 10 of a regular sized 2-liter soda bottle, right, is released into the interstitial space of the tissues every day due to capillary filtration, just normal filtration. That's an insane amount if you think about it. Once this filtrate is out of the bloodstream and in those tissue spaces, it becomes referred to as interstitial fluid. Of this, right, of those 20 liters, 17 of those are reabsorbed directly by the blood vessels. The lymphatic system drains that excess fluid and empties it back into the bloodstream via a series of vessels, trunks, and ducts. So lymph is the term used to describe interstitial fluid once it enters the lymphatic system. So once it moves from that interstitial space into the lymphatic vessels, it becomes known as lymph. Excuse me. When the lymphatic system is damaged in some way, such as by being blocked by cancer cells or destroyed by injury, this protein-rich interstitial fluid backs up, which results in it accumulating in the spaces between the cells of the tissue. And as you can imagine, there's no real estate for that to happen. So this inappropriate accumulation of fluid is referred to as lymphedema, and this can cause some very serious medical consequences. Unlike blood, lymph is not actively pumped by the heart. Instead, it is forced through the vessels by the movements of the body, by the contraction of the individual skeletal muscles during body movements, and by the lungs and the diaphragm moving during breathing. One-way valves, referred to as semilunar valves because their cusps, right, their little flaps, look like half moons, in these lymphatic vessels keep the lymph moving towards the heart and not backsliding, right? So you can see in the picture, right, backflow prevented by these valves. Lymph is transported from the lymph capillaries through successively larger and larger lymphatic vessels, referred to as lymphatic collecting vessels, until it finally is returned to the venous system through one of the two large ducts in the thoracic region. One is the right lymphatic duct, and this duct drains the lymph from the right arm and the right side of the head and thorax, or chest. The thoracic duct is the large thoracic duct that receives lymph from all the rest of the body. Both the right lymphatic and the thoracic duct empty the lymph into the subclavian vein on both sides of the body, on their respective sides, rather. So keep in mind that The subclavian vein is the one that runs right under the clavicle or collarbone. Now, the immune system is a collection of barriers, as you'll see. We talk about the skin, cells, and soluble proteins that interact and communicate with each other in very, very complex ways to protect us. The modern model of immune function is organized into 3 phases based on the timing of their effects. So phase 1 is referred to as barrier defenses, and these include the skin and the mucous membranes, right? The eyes, the mouth, the ears, the nose, the anus, the vagina, and act instantaneously to prevent pathogenic invasion into the body tissues. So those are sort of like the sentries at the guard. Phase 2 is the innate immune response, and this is a rapid but non-specific response, which consists of a variety of specialized cells and soluble factors, so factors that are just dissolved in the blood. And phase 3 is the adaptive immune response, which is slower, but very much more specific and arguably more effective in terms of the time that it takes and how efficient it is, which involves many cell types and soluble factors, but is primarily controlled by white blood cells known as leukocytes, right? Remember from the reproductive lecture, the corpus luteum, leuco is white. So white blood cells known as lymphocytes, which control immune responses. The cells of the blood, including all those involved in the immune response, arise in the bone marrow via various differentiation pathways from hematopoietic stem cells. So all those different types originally come from one stem cell, the hematopoietic stem cell, one type rather. We'll start with lymphocytes, which include B cells, T cells, plasma cells, which actually that should come right after B cells because B cells just mature into plasma cells, and cells called natural killer cells. So the 2 basic types of lymphocytes, B cells and T cells, are identical morphologically and are distinguished from each other by the surface protein markers that they express as well as the molecules that they secrete. While B cells mature in red bone marrow and T cells mature in the thymus, they both initially develop from the bone marrow. So B cells just happen to stay there and T cells move out. B cells are immune cells. They are lymphocytes, white blood cells, that function primarily by producing antibodies. An antibody is any of the group of proteins that binds specifically, like a lock and key, to pathogen-associated molecules. So molecules that are on specific microorganisms known as antigens. So an antibody is produced against an antigen. An antigen is a chemical structure on the surface of a pathogen, a disease-causing microorganism, that binds to T or B lymphocyte antigen receptors. Once activated by the antibody binding to its antigen, B cells differentiate into cells that secrete a soluble form of their surface antibodies known as plasma cells. So a B cell becomes activated and then transitions into a plasma cell. T cells do not secrete antibodies but perform a variety of functions in that same arm, the adaptive immune response. Different T cells have the ability to either secrete soluble factors that communicate with other cells of the adaptive immune response, or to destroy cells directly, those that are infected with intracellular pathogens or perhaps are cancerous. The roles of T and B lymphocytes in the adaptive immune response will be discussed in a few slides when we cycle back to the immune system. Okay, so this little table here just talks about the various types of lymphocytes and what their primary function is. Now, the primary lymphoid organs are the sites where lymphocytes mature, proliferate, right, divide and multiply, and are selected, which enables them to attack pathogens without harming the other cells of the body. And when we say selected, we'll learn in microbiology, they go through this really actually incredibly rigorous test, almost like you would train a dog. You want to test that it's not going to bite, so you give it certain like temptations or taunts and see that it doesn't respond. In a similar manner, the thymus will check that certain T cells don't respond against the body's own cells. When this check does not take place or it malfunctions, that's how we get autoimmune diseases. Okay, so the primary lymphoid organs are the bone marrow, where all the cells are produced and where B cells stay and mature, and the thymus gland, where T cells mature. There are also secondary lymphoid organs. So the lymphocytes develop and mature in the primary lymphoid organs, so the bone marrow and the thymus, but they mount immune responses from the secondary lymphoid organs. A naive lymphocyte is one that has left the primary organ and has then entered a secondary lymphoid organ. Naive lymphocytes are fully functional immunologically, but have yet to encounter an antigen to respond to, so they haven't been challenged yet. In addition to circulating in the blood and in the lymph, lymphocytes concentrate in secondary lymphoid organs, which include the lymph nodes, the spleen, and something called lymphoid nodules. The function of the lymph nodes is to remove debris and pathogens from the lymph. Any bacteria that infect the interstitial fluid are taken up by those lymphatic capillaries and transported to a regional lymph node. There, in the lymph node, immune cells internalize and kill many of the pathogens that are passing through, thereby removing them from the body. Sites of adaptive immune responses that are mediated by these B and T cells and the accessory cells of the adaptive immune system are heavily concentrated in these lymph nodes, so they're essential. The spleen is a fragile organ without a strong outer capsule that's attached to the lateral border of the stomach via something called the gastrosplenic ligament that we'll see in a few weeks. The spleen is dark red in color due to the fact that it's extensively vascularized because it's a filter, and again, sometimes called the filter of the blood because of its extensive vascularization and the presence of macrophages, which are professional phagocytosers, right? They eat the bad stuff, and dendritic cells that remove microbes and other materials from the blood, including dying red blood cells that need to be filtered out. Also, the spleen functions as the location of immune response to blood-borne pathogens. I apologize for the alarm there. The lymphoid nodules include the tonsils, the mucosa-associated lymphoid tissue, or MALT, and bronchus-associated lymphoid tissue, or BALT. The tonsils are lymphoid nodules located along the inner surface of the pharynx, or throat, and are important in developing immunity to oral pathogens. They're located at the back of the throat. Located at the back of the throat are the pharyngeal tonsils. These are sometimes referred to as the adenoids when they're swollen. I know when I was little they took out my quote-unquote adenoids or pharyngeal tonsils. Such swelling is an indication of an active immune response to an infection, meaning the body is actively fighting an infection, and they accumulate all sorts of materials taken into the body through eating and breathing, and they, quote-unquote, encourage pathogens to penetrate deep into the tonsillar tissues where they're acted upon by ready and willing cells sitting in those lymphoid follicles so they can be eliminated. Mucosa-associated lymphoid tissue, or MALT, consists of an aggregation of lymphoid follicles directly associated with our mucous membrane epithelia. The MALT makes up dome-shaped structures found underlying the mucosa of the gastrointestinal tract, our breast tissue, our lungs, and our eyes. Peyer's patches, which is a specific type of MALT in the small intestine, are especially important for immune responses against ingested substances, something that's been swallowed swallowed. I was going to say eaten or drank. Peyer's patches contain specialized endothelial cells called M cells, short for microfold cells, that sample material from the intestinal lumen, right, as it's passing over the intestinal epithelium, and transports it to nearby follicles so that the adaptive immune response can basically be primed and a defense can be proactively mounted against potential incoming pathogens. Bronchus-associated lymphoid tissue, or BALT, consists of lymphoid follicular structures with an overlying epithelial layer found along the bifurcations, aka the splits, the divisions from 1 into 2, 2 into 4, and so on and so forth, of the bronchi, and between bronchi and their surrounding arteries. They also have the typically less organized structure of other lymphoid nodules, and these tissues, in addition to the tonsils, are effective against inhaled pathogens specifically. MALT is for ingested pathogens. The immune system, kind of coming back full circle, can be divided into two overlapping mechanisms to destroy pathogens. The innate immune response, which is relatively rapid but nonspecific and not always the most effective, and the adaptive immune response, which is slower in its development during the initial infection, but is highly specific and much more rapid with every subsequent exposure to that particular pathogen. Barrier defenses, okay, any discussion of the innate immune system starts with speaking about the outermost physical defenses that are there to try to prevent pathogens from entering the body at all, to destroy them if they do enter slightly, or to flush them out in any way, shape, or form to prevent them at the end of the day from entering the bloodstream and really wreaking havoc and traveling throughout the body. However, barrier defenses are not a response to infections. They're just always there, working to protect against a very broad range of pathogens. The primary barrier to entrance of microorganisms is the skin. In fact, that outer layer that you learned about in A&P 1, where you see the stratum corneum, right? That's all dead keratinized epithelial cells. Those are too dry for bacteria to grow on them. And as the cells are continually sloughed off, they are carrying bacteria and other pathogens with them and away. Sweat and other skin secretions also lower the pH, right? Make it more acidic, contain toxic lipids, and physically wash away microbes. Saliva is rich in lysozyme, which is an enzyme that destroys bacteria by digesting away their cell walls. The acidic environment of the stomach is fatal to many pathogens, and the mucus layer of the GI tract, the respiratory and reproductive tracts, and then that mucous membrane of the eyes, ears, and nose, both traps microbes in the mucus and traps other debris, but then also with cilia facilitates their removal. In terms of the cells of the innate immune response, we have the phagocytes, which is a catch-all term for the certain types of cells that work to engulf, right, to ingest or eat microorganisms inside of a structure called a phagosome. So they basically contain a bag of lysosomal enzymes. Enzymes, think of like Lysol. And then they pull in the microorganism in a phagosome. And those two membrane-enclosed sacs fuse to form the phagolysosome, in which many pathogens are killed. The two phagocytes that we focus on, the large majority, are macrophages and neutrophils. Macrophages are irregularly shaped phagocytic cells, or phagocytes, that are amoeboid in nature. They remind me of those little toys that used to be at the checkout that are like just a sack full of like gel. Every time you go to squeeze them, it like oozes out of your hand. So they're amoeboid in nature, and they're the most versatile of the phagocytes in the body. They can move through tissues and squeeze through the little cracks in the single-celled walls of capillaries using pseudopodia. They basically project out a section of cytoplasm to make these like little false feet. They not only participate in the innate immune response, but they also have evolved to cooperate with lymphocytes that are part of the adaptive immune response, the B and T cells. They help, they bring things to them. Neutrophils are the most common type of white blood cell in a healthy individual that are just circulating around. They're kind of just ready and waiting. And they're attracted via something called chemotaxis. So when there's an infected tissue, they move towards, right, taxis movement, towards the chemicals of the inflammatory response. These somewhat spherical or round cells are granulocytes, meaning that they contain these little cytoplasmic granules, which in turn contain a variety of what we call vasoactive mediators. So things like histamine, if you think of like if you get a bug bite, the blood goes to that region, it swells, it gets red, it gets itchy, right? In contrast, macrophages, the other type of phagocytes, they're agranulocytes. They don't have the granules in their cytoplasm. There are also natural killer cells, and this is a type of lymphocyte, white blood cell, that has the ability to induce apoptosis, or programmed cell death, in cells that are infected with intracellular pathogens. So cells that are infected with like an obligate intracellular bacterium, such as rickettsia, right? The bacterium that causes, oh my goodness, typhoid, or Borrelia burgdorferi, the bacterium that causes Lyme disease. Those actually get inside of our white blood cells, the cells that are there to defend against them, and so we have to destroy them, right? Or a cell that's been infected with a virus. If you think about it, it's kind of like a security center has been infiltrated. There's way more inside and we can't get them out. So what do we have to do so that it doesn't keep spreading? You just kind of kill the cell that they're in. Now, the job of the innate immune system, we've said repeatedly, is it's fast-acting, but it's non-specific. So the cells of the innate immune response, the phagocytic cells, and those cytotoxic natural killer cells work by recognizing sort of general patterns of pathogen-specific molecules, such as certain components of the bacterial cell wall or particular flagellar proteins, by using something called pattern recognition receptors. A pattern recognition receptor, or PRR, is a membrane-bound receptor that recognizes certain characteristic features of a pathogen, as well as molecules that might be released by stressed or damaged cells. So those receptors, or excuse me, those pathogenic molecules, the pattern recognition molecules, are not specific to one particular species of bacteria, but possibly to, say, a number of different species within a certain genus, or maybe a few different genera of bacteria. They're not particularly specific. So these receptors, which are thought to have evolved prior to the adaptive immune response, are present on the cell surface whether they're needed or not. However, as I've said, their variety is very limited, which means that the innate immune response must sort of get by using only a limited number of receptors that are active against as wide a variety of pathogens as possible. This is opposite to the adaptive immune system, which uses large numbers of different receptors, each which are highly specific to a particular pathogen. I'm going to just talk briefly about the complement system. We could spend an entire lecture on this, so keep in mind this is just an overview. The complement system is a series of proteins constitutively, meaning constantly, found in the blood plasma, the fluid portion of the blood. And the complement system works with the inflammatory cells of the innate immune system, as well as those of the adaptive immune system, by binding to the cell membrane of a pathogen that activates it, and in doing so basically labels it for phagocytosis. This labeling process is known as opsonization. It diffuses away from the pathogen and acts as a chemotactic agent to attract or draw in phagocytic cells to the site of inflammation, and they also can work different members of the complement system to form damaging pores in the plasma membranes of a pathogen so that they basically spill their guts out and die. The inflammatory response is the hallmark of the innate immune response. The release of damaged cellular contents into the site of tissue injury is enough to stimulate the inflammatory response. The inflammatory reaction brings in phagocytic cells because it releases chemicals or produces chemicals that attract them through chemotaxis so that they're called over to clean up the scene. Now, the adaptive immune response, in contrast to the innate, is very, very specific. It's able to specifically recognize and make a response against a wide variety of pathogens. Antigens, which is the small chemical group associated with particular pathogens, are recognized by specific receptors on the surface of B and T lymphocytes. The adaptive immune response to these antigens is in fact so versatile that it can respond to nearly any pathogen. Knock on wood, right? So let's first talk about the primary adaptive response. when the immune system is first exposed to a pathogen, so the first time, it mounts a primary adaptive response. Symptoms of a first infection, called primary disease, are always relatively severe because it takes time for the body to develop an initial adaptive response that is more effective. It becomes more effective over time. Upon re-exposure to the same pathogen, a secondary adaptive immune response is generated, which is stronger and faster than the primary response. This secondary response, I realize I should have changed the title of this slide to secondary, is the basis of immunological memory, which protects us from getting the same diseases over and over again. Now, you may recall during COVID, albeit that was a virus, so different than a bacterium, but it could change the spike proteins on its surface. So the problem why people were concerned with it mutating, right, same thing with the flu, influenza, is that if the antigens change, then that initial adaptive immune response is sort of negated, right? It has to start all over again in building. Here you can see this is a very iconic representation of the primary versus secondary immune response and overall how the adaptive immune system works. You have an initial exposure to a pathogen, and you see there's sort of not steep incline where we mount that primary immune response over time. And you see the concentration of a particular antibody in the blood is around, they don't put a scale, right, but it's much lower as opposed to the secondary exposure to that exact same pathogen. That's the caveat. It has to be the same pathogen. You see that the immune response is way faster and way more intense. Antigens, as we've said, are surface proteins. They can be lipopolysaccharides and or peptidoglycans on the surface of a cell most of the time. And it's a marker that tells your immune system whether something is harmful or not, whether it's self or other. Antigens are found on viruses, on bacteria, on tumors, and on normal cells, just identifying self as self. The B cell adaptive immune response is in reaction to particular antigens, right? It produces antibodies. So B cells work with other cells in our immune system to fight harmful invaders that can make you sick, as well as abnormal cells like cancer cells, anything that identifies as non-self. Once B cells are activated, they become plasma cells that then actively produce antibodies in response to one particular antigen. Or they become memory B cells that basically are primed and waiting so that the second time, third time, fourth time you're exposed to that same pathogen, they're ready to go. It's why the immune response the second and every subsequent time after that is stronger and faster. An antigen-presenting cell, known as an APC, attaches to an antigen, breaking that antigen down into smaller pieces. The antigen-presenting cell then attaches the antigen pieces to a molecule on its surface called the major histocompatibility 2 complex, or the MHC2 complex. Helper T cells, which is one type of T cell, binds to these surface MHC2 complexes. This binding activates the helper T cells, and they go over and then spur into action the B cells. An activated helper T cell attaches to a B cell, causing it to make copies or clones of itself. Some of the B cells become plasma cells capable of producing antibodies. Others, as I've said, become memory cells that get stored in the body. Plasma cells make millions upon millions of antibodies over the next several days. All antibodies are customized to destroy only that one specific pathogen, and these antibodies then prevent the pathogens from causing further harm to the body. There are also cytotoxic T cells. So cyto means cell, toxic means poisonous or harmful. So these kill directly in cells that are infected with viruses and bacteria or tumor cells in some cases. Whereas, as I've already said, the helper T cells, they don't kill cells directly, but they stimulate B cells to produce antibodies. They can also signal for the cytotoxic T cells to come over and quote quote, do the dirty work. Each T cell is unique in that it's designed to fight only one type of intruder. Once your immune system identifies the threat, it locates the specific T cell that's been designed to combat that particular pathogen. So the T cell copies itself, makes more T cells, just like, right, any part of the adaptive immune response, you're going to see a common thread. Not only do they ramp up in a much more specific manner, but they also keep knowledge for the next time that something happens. So these effector T cells, or cytotoxic T cells, join the fight in conjunction with the helper T cells and the B cells. Your T cells continue to protect you even after the intruder is gone, as I said, because some of these, just like B cells or plasma cells, become memory cells. Unlike effector T cells, the memory T cells aren't fighters, but they remember the intruder so that if it returns, the immune system can mount a very quick and effective response. And a cell called an antigen-presenting cell, or an APC, locates evidence of the intruder and attaches it to a complex called a major histocompatibility, or MHC complex, on its outer surface. This step is important because T cells can't recognize evidence of an intruder unless it's attached to a major histocompatibility complex. The T cell binds to the MHC, and there are two types of MHCs. One fits each type of T cell. So on a cytotoxic T cell, it has a CD8 receptor, which can only bind to MHC1 molecules. The helper T cell has CD4 receptors, which bind to MHC type 2. Once the T cell binds with all the matching parts, it becomes activated. Okay, so here we have an immune cell. It's either part of the innate immune cell system or acquired, and then you can see the various types with dendritic cells and this type, which we don't talk about, the gamma delta T cell, in the middle. And here just a few summary slides which take into account everything we've talked about and just put more detail onto the graphics. So you see what's happening at each phase of the primary and secondary immune response. And this is just, I like this slide because it talks about immunity, right? Acquired immunity, well, we have adaptive immunity, and it can be naturally acquired or artificially acquired. And this breaks it down two different ways. Active immunity can be natural, right? You literally get infected with something and the body has to naturally and actively fight it. Or artificial. You get a vaccine and your body mounts an immune response against that to condition itself for the next time. Or there's passive immunity. So a mother secreting antibodies in her breast milk to a baby is a passive immunity, as well as artificial. Some people are actually missing certain, say, complement factors or antigens, so they're given these intravenously. Okay, and then as far as different ways to make a vaccine, there are inactivated vaccines which use a killed virus, a dead virus, to trigger an immune response because it still has the surface proteins that are needed to activate the immune response. There can be attenuated, where they're alive, but they're a weakened version. There are subunit vaccines, which take, usually with a virus, just take a tiny portion of it, or a nucleic acid vaccine. So for COVID, there were both the nucleic acid vaccines as well as the inactivated. Okay, so that is it for